Hello viewers, welcome to my channel Biology Notes. Today I will discuss about excision repair of damaged DNA. This repair involves at least 3 steps. In step 1, a DNA repair endonuclease or endonuclease containing enzyme complex recognizes, binds to, and excises the damaged base or bases in DNA. In step 2, a DNA polymerase fills in the gap by using the undamaged complementary strand of DNA as template. In step 3, the enzyme DNA ligase seals the break left by DNA polymerase to complete the repair process. There are two major types of excision repair: base excision repair and nucleotide excision repair. Both excision pathways are operative in the dark and both occur by very similar mechanisms in E. coli and humans. So first I will discuss base excision repair mechanisms. After DNA proofreading by DNA polymerase, base excision repair is the most important mechanism used to remove incorrect or damaged bases from DNA. The main target of base excision repair is non-bulky damage to bases. This type of damage can result from the variety of causes, including methylation, deamination, oxidation, or the spontaneous loss of a DNA base. Now, see this diagram. This is a double-stranded DNA. In the next step, this cytosine get deaminated and changed to uracil. So, this uracil is an abnormal base and needs to be removed. A group of enzymes called DNA glycosylases recognize abnormal bases in DNA. Each glycosylase recognizes a specific type of altered base such as deaminated bases, oxidized bases, and so on. So here abnormal base uracil is detected by uracil DNA glycosylase. In the next step, the glycosylases cleave the glycosidic bond between abnormal base and sugar. As a result, apurinic or apyrimidine sites or AP sites are produced. AP sites are recognized by enzymes called AP endonucleases. AP endonuclease then nicks the damaged strand upstream of the AP site. AP endonuclease with deoxyribophosphodiesterase help to excise the sugar phosphate groups at these sites. DNA polymerase then replaces the missing nucleotide according to the specifications of the complementary strand. Here it is cytosine. Finally, DNA ligase seals the new nucleotide into the backbone. So, from the discussion, we come to know that DNA glycosylases play the most important role in base excision repair. These enzymes were first discovered in bacteria and have since been found in all kingdoms of life. There are two main classes of glycosylases: monofunctional glycosylases and bifunctional glycosylases. Monofunctional glycosylases have only glycosylase activity, whereas bifunctional glycosylases also possess apelase activity that permits them to cut the phosphodiester bond of DNA, creating a single strand break without the need of an AP endonuclease. Here I prepare a table showing few examples of glycosylases which are found in E. coli, yeast and humans. So you pause the video, see the table for your better understandings. Problems of base excision method. It cannot correct bulky adducts such as the cyclobutane pyrimidine dimers caused by UV light. Such bulky adducts distort the DNA helix. It cannot correct damage to more than one base. So this is all about repair of single strand damage of DNA by base excision mechanism. If you have any query, please comment. Share the video if you like. Subscribe the channel for more new videos. Stay with me. Keep watching. Thank you.